Good morning, class. Today we're going to talk about mutations in DNA. So up until now, we've said that A always pairs with T and C always pairs with G, as long as we're talking about DNA. Today we're going to talk about what happens when this goes wrong, and this is called a mutation. So mutations are permanent changes in the cell's DNA, and it changes either the structure or the amount of DNA that we're seeing. So let's say the original strand is this top one right here, and we have a change in the bases. So this is supposed to be A pairing with T, but now all of a sudden the incorrect base was paired with it. So now A, when it's being copied, is being paired with C. And so that new strand of DNA is going to be incorrect. And that change is permanent. There's nothing that can be done to fix it after the fact. We could also see other changes like deletions, and we'll talk about those here in just a second. But the change to the DNA is permanent, and it's typically made during the copying or creating of a new cell or a new strand of DNA. So some causes for mutations. I bet we've heard of a few of these. But the most common one actually is just a mistake when copying the DNA. So the cell is unzipping that DNA and trying to make a copy, and it just puts in the wrong base pair. But there can also be environmental factors that cause mutations as well. So there are mutagens, which are things that we know cause mutations to occur. So radiation is one of those. RV radiation specifically that comes from the sun, we know is bad for us. We're supposed to put on sunscreen, but maybe we don't always. And exposure to this radiation over time can lead to mutations within our DNA, specifically within our skin's DNA. X-ray exposure. Anyone ever had an X-ray before? You're only supposed to get so many within a short period of time, so doctors keep track of how many X-rays or how often they're doing X-rays on you. A lot of times you see this with like the dentist's office. They do it every year, maybe every other year, but you're not getting a whole bunch within 1 year because exposure to too many X-rays can lead to mutations. There are certain chemicals that we intake that we know or that we believe cause mutations within our DNA as well. Cigarette smoke. A lot of us have heard that cigarettes cause cancer. And that cancer is caused by mutations that are occurring within our body because of chemicals that are put into those cigarettes. A lot of the preservatives that we use in food and our processed food can cause mutations. And then some of our acne products contain a chemical ingredient that can also cause mutations as well. So again, it's not just a one-time use, but it's over many exposures to these chemicals or to this radiation that can cause mutations to occur. Other ones could be infectious agents. So we've talked about HPV, a sexually transmitted disease, or some other different types of bacteria that can cause mutations to occur within our DNA. So all of these would be causes of DNA. Now, once this causes of the mutation of our DNA. Once these mutations occur, there's basically two different ways that this can happen. So you can have mutations that could actually be beneficial, or you can have mutations that are detrimental, which are bad for us, and that's most of the time what we see are detrimental disorders. So there's lots of mutations that we see up here. If you see, like the fly, for example, a mutation that causes them to be wingless. Now, benefit or detriment? If you think about a fly, if they don't have wings, they can't escape a predator. So this is definitely a downside, probably actually going to lead to death for this individual. So that would be a detrimental mutation. The mouse with no fur may also be a detrimental mutation. With no fur, it's not able to keep warm, or it can have skin irritations easier. So maybe it won't kill it, but it's definitely a disorder or something that's going to make their life a little bit more difficult. Now, if we look at the rabbits in the top right-hand side, one's brown and one's white. The mutation is occurring in the white one, where they're not able to produce pigment in that fur. Now, in this background right here, the white fur may actually be a detriment. But if I were to put that rabbit in snow, that may actually be what we call an adaptation or a beneficial mutation. And we'll talk about that more in evolution. But mutations, they can either be good for us or bad for us. But most of the time they are bad for us. And we do not get to pick the mutations that occur. They just happen, normally by accident or by environmental exposure. So if we look at specific types of mutations, the types of mutations that we can see when we're looking at the actual letters or the bases inside of the DNA, there's 3 different types. We can have a point mutation, an insertion, or a deletion. So a point mutation is if I were to point to one specific base pair, like one specific location on that DNA, and it's supposed to be CG, but instead now it's TA. It's completely changed. And it may not seem like that big of a deal, but those letters are what make up the difference between you and me and between me and everything else, right? So the letters carry specific instructions for proteins to be made, and when we change any of those, it changes the protein that's going to be made. So that can be a big change that we see. The protein isn't able to be produced correctly or functional, or it's maybe making the wrong protein. So point mutation, changing a single base pair. An insertion, on the bottom right-hand corner, is where we see an extra base added in, or maybe more than one base. And again, as we add in one or more bases, this is going to cause what we are going to call a frame shift. If we think back to where the proteins are made in sets of 3, those codons, now when we read this, it doesn't just go TAA-CTG, it now goes TAA-CCT. It completely changes the proteins and the codons and how that's going to be read. Same thing happens with a deletion, where we're going to remove one or more bases. And again, the more information you add or remove, the more impactful that mutation can be, and probably detrimental. So types of mutations, we've kind of talked about this. That frame shift with an insertion or a deletion, it's going to cause a change in how we read that DNA, since we read it by 3 letters at a time. A silent mutation means that there was a change in the letters, like the bases, but it actually didn't change the protein that we're making, so we don't see any effect in it. And in fact, in every single human, there's probably several silent mutations that are happening, but it causes no good or bad effect because we're making the same protein at the end of the day. The only other one that I want you guys to know about on this slide is a missense mutation or a replacement mutation. Basically, the wrong codon or the wrong amino acid is getting added in. So that's it from this slide. And then mutations, when we get to the larger level, and we're talking about a large section of DNA. Since DNA makes up chromosomes, and we kind of like twist and scrunch it together to fit into a chromosome, when we see chromosomal mutations, these are normally severe, like extremely impactful, typically past a certain point are going to be fatal for whatever organism they're occurring in. So there's 4 different types of chromosomal mutations. The first one is a deletion, and this is where a section of the chromosome is lost or it's deleted. So think, if we've got DNA that's being twisted and scrunched down into this, that's a huge section of information that is lost. Sometimes it's not as important, but normally chromosomal mutations are pretty impactful. Duplication means extra information is added in, and again, you would think, well, what's the big deal because the information's still there. But when we're trying to process, or when we know that if we go exactly to this one location and I can find height or hair color, and now I'm reading something different, it can be like you've lost that information because you don't know where it's located at. So that can be very impactful for the organism. Duplication, chromosomes, or duplications of sections of chromosomes can lead to some very severe disorders. Inversion is the flipping of a piece of the information upside down, basically, or exchanging that information kind of backwards. Again, when we know to look in a specific location or at a specific section of a DNA chromosome for a certain trait like eye color, hair color, then we don't see it there, or if we don't see it there, we think it's lost. So even though the information is still contained within the chromosome, we're not able to see it or find it. Translocation is even worse. This is where our information isn't even just lost in the same piece of chromosome. It's actually exchanged with a different chromosome or a different area. So this chromosome, there's no just continuing to search down the line for this information. It's completely misplaced into a very different part of our karyotype, if you remember that. So like maybe it's supposed to be on chromosome 1 and it's now being placed on chromosome 15. There's going to be no finding of that information. So again, to go back and repeat just a little bit, mutations can be good or bad or silent maybe. Most of them are bad, and we don't get to pick which ones we get. They happen by mistake or by environmental exposure, and normally they result in illnesses, potentially death. And then when we're talking about chromosomal mutations, these are severe, and a lot of times they're not survivable. So that's what you need to know. You guys are now going to watch a short YouTube video before working on a DNA to disease worksheet where you guys get to play doctor today and diagnose the mutation that has happened in your patient. Please email me if you have any questions. Thank you.